Hello everybody, my name is Promit Kumar Chaudhary and currently I am a professor in the Department of Chemistry at IIT Delhi. Before I share with you some details about this course on biophysical chemistry, I will just give you a brief introduction about myself. As I said, I am presently in the Department of Chemistry, IIT Delhi, and my group mostly works on experimental physical chemistry, and the research that we are involved in is focused on the behavior of proteins in crowded environments. Do realize that the interiors of cells are very crowded. So, we try to look at how crowded environments affect protein conformations and dynamics using a host of spectroscopic techniques, out of which fluorescence is the one which we use the most. That is, fluorescence spectroscopy is the one which we mostly use, and this is something which we will also discuss in the course later. Now, coming to some details, a brief overview about this course. As you can see, the title of this course is Biophysical Chemistry, and when we think about this topic biophysical chemistry, then immediately what we try to look at or kind of connect to is the definition of this particular area. So, if you go to the next slide, as you can see, the title of the slide says what is biophysical chemistry. So, we can approach the concept of biophysical chemistry in a couple of ways. The first definition, this involves the study of biological macromolecules by applying the principles and concepts from the areas of physics, chemistry and biology. To put it in a different way, but having the same meaning, if you go to an alternate definition, we can say that it is an application, or this biophysical chemistry is the application of the basic tenets or principles of physical chemistry to problems in biology. To say the least, if you can understand or realize by these definitions that the scope of this topic is indeed very broad, and a single course like this where we will be having about 42 lectures probably will not be able to cover everything. It is virtually impossible. So, in the next slide, what I am going to share with you is a brief outline of the topics that we are going to cover in this course over the next 42 lectures. So, as I said, the brief outline, the first topic being ribosome and protein synthesis. Now, ribosome and protein synthesis, as the name is suggesting, ribosome is a place where a protein is synthesized, and so we look at different aspects of the same. Then we look at structure of proteins, because this is extremely essential, as many of you would be knowing that the proteins have different levels of structure, starting from the primary structure, which is what? The sequence of amino acid residues in a particular protein, then the secondary structure, then the tertiary structure, and then the quaternary structure. We will look at some details in the structural aspects and also different forces that hold the native protein together in its compact structural form. Following this, we would go and discuss, we will go to rather and discuss about the principles and models of protein folding. Now, what does this mean? What it means is, so when the protein is synthesized from the ribosome, that means the amide bonds are formed and the protein is synthesized by linking up the different amino acids. At that time, the protein does not have much structure. However, once the protein is released into the cytosol, then the protein starts developing structure. This is where you come in and ask, what are the different pathways or models by which a protein can fold from its primary structure, that means its disordered structure where it does not have much structure, to its final native state, where it has a compact 3-dimensional structure. Then we will move on to the thermodynamics of protein folding. So, thermodynamics of protein folding has to deal with the stability of proteins, and again different aspects that have an effect on the stability. This will be followed by kinetic studies. So, as kinetics mean, that means we are talking about what the rate of process, how fast a process is happening. So, the same here, from the denatured state of the protein to its final native state, how much time does it take? That means, how fast can the protein fold? And this would be the last topic that we will be covering in the course. This has to deal with techniques used in the study of protein folding, with some emphasis on instrumentation wherever applicable. So, as you can see that wherever or whatever topics have been outlined, almost every topic has the word protein in it. Thus, this course is mostly about protein structure and dynamics, as mentioned by the topics in the slide. And to end this brief introduction about the course, these are some of the books that I will be following and will be discussing from over the entire course. So, you are very much encouraged to look these books up. These books are very good books, and they have many details about the different aspects that we are going to touch in this course. So, I hope you enjoy this course, and again, if you have any problems during the entire course, even small things that you do not understand, you might be facing difficulties with, please do not hesitate to contact me by email. Again, I will go to the slide, the first slide where my email address is written under my name. So, please do contact me if you have any issues with regards to the basic understanding of the topics in this course. Again, thank you, and I hope you like the course.